University of British Columbia researchers have found a cheap, sustainable way to build a solar cell using bacteria that convert light to energy. Their cell generated a current stronger than any previously recorded from such a device and worked as efficiently in dim light as in bright light. This innovation could be a step toward wider adoption of solar power in places like British Columbia and parts of northern Europe where overcast skies are common. These solar cells are called as biogenic because they are made of living organisms. With further development, they could become as efficient as the synthetic cells used in conventional solar panels. Solar cells are the building blocks of solar panels. They do the work of converting light into electrical current. Previous efforts to build a biogenic solar cells have focused on extracting the natural dye that bacteria use for photosynthesis. It is a costly and a complex process that involves toxic solvents and can cause the dye to degrade. The UBC researchers' solution was to leave the dye in the bacteria. They genetically engineered E. coli to produce large amounts of a dye that gives tomatoes their red-orange color and is particularly effective at harvesting light for conversion to energy. The researchers coated the bacteria with a mineral that could act as a semiconductor and applied the mixture to a glass surface, with the coated glass acting as an anode at one end of their cell. They generated a current density of 0.686 milliamps per square centimeter, an improvement on the 0.362 achieved by others in the field.